I am going to discuss on the topic comparative account of the brain. The brain is the main part of the central nervous system. It is a center for all senses and muscular coordination. The brain is enclosed in the cranium and covered by the membrane that is called the meninges. And the meninges consist of three layers. That is a outermost layer. The outermost layer that is known as the top layer, that is a dura mater. And then middle layer, the middle layer that is known as the arachnoid, and the pia mater, that is a innermost layer, and that contains the blood vessels. And meninges act as protective function. In all vertebrates, the brain develops from embryonic ectoderm. The early developmental pattern is very similar in all vertebrates. If you see the structure of a brain in all vertebrates, the main divisions of the brain are the forebrain, midbrain, and the hindbrain. Forebrain is also known as the prosencephalon. Midbrain is also known as mesencephalon, and the hindbrain is also known as rhombencephalon. The prosencephalon is further divided into anterior telencephalonic part and the posterior diencephalonic part. The main part of the prosencephalon that is known as cerebrum. Besides its prosencephalon consists of olfactory lobe, olfactory peduncle, pineal body, thalamus, hypothalamus, etc. Whereas the mesencephalon that remains undivided, it contains paired optic lobes. The rhombencephalon consists of metencephalon and the myelencephalon. The metencephalon forms the cerebellum. The prosencephalon forms cerebrum, whereas the metencephalon forms cerebellum. While the myelencephalon that forms medulla oblongata, which constitutes as the spinal cord. The cavities of the brain that is known as the ventricle. There are four ventricles present in the brain. The first and second ventricle, that are the lateral ventricle, that are present in the cerebrum, and the third ventricle that is present in the diencephalon, and the fourth ventricle that presents both inside the metencephalon and myelencephalon. First and second ventricle that connected to third ventricle separately by the foramen of Monro. Whereas the third ventricle that communicates to the fourth ventricle by narrow canal, that canal is known as iter or aqueduct of Sylvius. The cell body of the neurons become segregated in central part of the neural tube, which persists in spinal cord region. In the brain region, the cell bodies migrates towards the periphery. Thus, gray matter and white matter are differentiated. Here you see the different parts of the brain. Basically, the brain has three main divisions: prosencephalon or the forebrain, mesencephalon or the midbrain, and rhombencephalon and the hindbrain. The prosencephalon is further divided into anterior part. That part is a telencephalon that is differentiated into olfactory tracts, cerebral hemisphere, corpus stratum, and hippocampus. Whereas the diencephalon differentiated into thalamus, hypothalamus, and the pineal organ. And mesencephalon that remains undivided, optic, auditory lobes, you see here, and the cranial nerves, third and fourth origin from the mesencephalon. And the hindbrain, rhombencephalon, that is differentiated into metencephalon and the myelencephalon. The metencephalon that's from cerebellum, that is a cerebellum, and that is the pons. Whereas the myelencephalon that forms the medulla oblongata, which constitutes the spinal cord, and the cranial nerves 5 to 12 originate from myelencephalon. That is a basic organization of the brain. Now, the cerebral hemisphere derived from the telencephalon, that is mainly associated with intelligence, thought, reception, and conduction of smell. At the anterior end of cerebral hemisphere is an outgrowth that is known as olfactory lobe and subsequently associated with the posterior part of the olfactory apparatus. The cerebral hemisphere enlarges to an increasing degree as the vertebrate scale is ascended from fish to mammal. The floor of the cerebral hemisphere is differentiated into corpus stratum, that is a reticulated structure, and the remainder part that is known as the pallium. The pallium also become highly developed and modified in the evolution of higher group of vertebrates. Now, the telencephalon and cyclostomes. In cyclostome, each hemisphere is divided into an anterior olfactory bulb and a posterior olfactory lobe. It concerned with receiving impulses from the olfactory apparatus and relaying them to the diencephalon. In fish, the telencephalon consists of a pair of solid olfactory lobe in cup, whereas it is rudimentary. And then cerebral hemisphere. The cerebral hemisphere are in the form of the solid mass joined with each other in the midline, and that is covered with thin wall pallium that is non-nervous. Olfactory lobes are conjoined with the olfactory bulbs by olfactory nerve. The narrow space between the roof and the solid hemisphere may be considered as first and second brain ventricles. In elasmobranch and dipnoans, the pallium is fairly thick. In fish, telencephalon also act as an olfactory center. In amphibia, two olfactory lobes form the anterior most part and emerge almost imperceptibly with the anterior end of the cerebral hemisphere. The pallium is thicker in comparison to fishes and some cells from the gray matter and that have moved peripherally. The pallium is divided into two general regions, a dorsal medial that is known as the archipallium and the more lateral that is known as the paleopallium. Both these structures are related to olfactory sense. In reptiles, the cerebral hemisphere have increased in size and grown backward to cover partially the diencephalon and separately in the medial portion by the deep fissure. The olfactory lobes are long and connected to the cerebral hemisphere by a narrow peduncle. In certain reptiles, the new pallium has appeared at the anterior dorsal end of each hemisphere between the archipallium and paleopallium area. In crocodilians, for the first time, nerve cells migrate into the neopallium and become arranged along its outer surface, and that forms a true cerebral cortex, which serves as the association center. In birds, olfactory lobes are practically rudimentary. Archipallium and paleopallium are also present, but neopallium is lacking in most forms, so there is no cerebral cortex. The cerebral hemisphere are large because the corpus stratum is of unusual size. In mammals, the cerebral hemisphere is very large as neopallium has increased enormously, that pushing archipallium area medially and ventrally, where it continues to serve as an olfactory center. The paleopallium is pushed ventrolaterally, where it becomes piriform lobe. It also acts as an olfactory lobe. Piriform lobe is separated from the neopallium by rhinal fissure. That is a rhinal fissure. The cerebral cortex is best developed in man, where it is a few millimeters in thick. Archipallium is pushed by neopallium, moves roll on itself lengthwise and sinks largely below the surface and forming a long arching band that is known as hippocampus. Its name is suggested by its rolling appearance seen in cross-section. Hippocampus is associated mainly with memory. The basal nuclei are moderately well developed into the interior of the hemisphere, as the corpus stratum divided into amygdala, that is the archeostratum, globus pallidus, paleostratum, and neostratum, that is caudate nucleus putamen. The mammalian corpus stratum is less conspicuous. The surface of the cerebral hemisphere become folded and convoluted so that ridges and depressions appear. The ridges are also known as gyri and the depression are known as sulci. Larger mammals have more convolution than smaller species. Starting from marsupials, a broad white mass, that is corpus callosum, which composed of medullated fibers that connects to cerebral hemisphere. Now come to another divisions of the forebrain, diencephalon, that is also known as thalamencephalon also. And the sensory portion of the eye developed from the diencephalon. It is a small structure which is entirely hidden by enlarged cerebral hemisphere, and it is divisible into dorsal epithalamus, lateral thalamus, and ventral hypothalamus. It is divisible into epithalamus, thalamus, and hypothalamus. The epithelial lining of the dorsal roof plate covering the third ventricle, that is a diocele, together with pyometer, that's from telachoroidea. That's from telachoroidea. Now the vascular folds of the telachoroidea extends into the third ventricle, that from the anterior choroid plexus. Now, posterior to the telachoroidea, the roof of the diencephalon consists of the epiphyseal apparatus. In several lower forms, it is composed of anterior parapineal or parietal body, posterior pineal body or epiphysis, although both are present in lamprey, some frogs, some fishes, sphenodon, and numerous lizards. Only the pineal body has persisted in most fishes, urodels, many reptiles, birds, and mammals. However, highest degree of development of parapineal body that occurs in the sphenodon. That's from the median eye. It is developed to lesser degree in some lizards. An interparietal foramen in the skull provides passage for nerve connecting the median eye and the brain. In tadpole stage, pineal body is connected to the pineal stalk, but in adult frog, it is separated from the pineal stalk and lies outside the skull below the skin at the brow spot. In case of lamprey, both parapineal and pineal organs are associated with the eye-like structure that having retinal lens. In all vertebrates, the pineal body is present and glandular in nature. The gland has a connective capsule with septa extending inward. The parenchyma is made up of pinealocytes and glial cells. The pinealocytes secrete melatonin that is involved in daily and seasonal photoperiodically induced rhythms in sexual behavior and reproduction and thermoregulation also. Melatonin synthesis and secretion are increased during the dark period of the day and maintained at low level during the daylight hours. Thus, it controls the light and dark cycle in the environment. However, in the course of evolution, there has been a change from a primitive photoreceptive type of organ to a secretory structure. So, the diencephalon, you know, that's divided into dorsal epithalamus, lateral thalamus, and the ventral hypothalamus. Epithalamus is a dorsal segment of the diencephalon. It includes the habenula and their interconnecting fibers, the habenular commissure, strata medullaris, and pineal organ. The function of the epithalamus is to connect the limbic system to other parts of the brain. Now, the ventrolateral wall of the thalamus that consists of a large number of nuclei that are very important relay center. Significant center are the lateral medial geniculate bodies, which relay optic and auditory impulses respectively. In reptiles and mammals, the walls are thickened inwardly to meet the center of third ventricle by a mass of gray matter that is known as intermediate mass or soft commissure. The thalamus play an important role in regulating states of sleep and wakefulness. The ventral portion of the diencephalon attains its maximum development in the form of hypothalamus and infundibulum. The hypothalamus contains nerve center which integrate the function of the peripheral autonomic system, which those of other nerve tissues. The important releasing factor and releasing inhibiting factors of the hypothalamus that affect the release of hormone by the anterior lobe of the pituitary gland via the hypophyseal portal system. Hypothalamus consists of optic chiasma, where the optic nerve process, tuber cinerum, believed to be the parasympathetic center, a pair of mammillary body for integration of olfactory cells, infundibulum, the distal portion that contributes to the posterior lobe of the pituitary gland. Now come to mesencephalon. The embryonic midbrain undergoes relatively less changes than other portion of the brain. The floor and the side wall of the mesencephalon that composed of fiber tracts, the cerebral peduncles that connecting forebrain and hindbrain. Actually, mesencephalon consists of dorsally placed optic tectum and ventral tegmentum. The tectum is thick than tegmentum because of accumulation of gray matter. In lower vertebrates, tectum is the form of two well-developed optic lobes. Two well-developed optic lobes or corpora bigemina, which serves as center for the visual sense. Each optic lobe contains a large cavity that is an optic ventricle, two ventricles together forming the mesocele. In higher vertebrates, optic lobes are almost solid structure, and only the narrow cerebral aqueduct passes through the center of the mesencephalon, and a transverse fissure divides the optic lobe into four prominences. Corpora quadrigemina, that is a tectum, the anterior part, that is a superior colliculi that contains a receptive center for the visual sense, and the posterior part, inferior, that is a posterior colliculi that serves to integrate auditory impulses. The superior colliculi are less important as visual center with the great development of the cerebral cortex. The optic lobes are more conspicuous features of the brain of lower vertebrates and birds. In mammals, the superior colliculi have become less important as visual center with great development of the cerebral cortex. Now come to metencephalon. The cerebellum develops as a large dorsal outgrowth from the metencephalon. Its function is to coordinate the neuromuscular mechanism of the body. The ventral portion of the metencephalon in lower vertebrates is composed of heavy fiber tracts which emerge with medulla oblongata. In fish, cerebellum is partly visible externally as corpus cerebelli, and its anterior part enter into the cavity of optic lobes that's from the valvula cerebelli. That is, you see here, valvula cerebelli. Now, the posterior, the corpus cerebelli consists of the outer molecular layer and the inner granular layer. The corpus cerebelli, if you see the corpus cerebelli that you see here, outer layer, that's the outer layer, that is a molecular layer, and the inner layer, that is a granular layer. And if you see the valvula cerebelli, then just reverse in position. The cavity of the cerebellum, that is known as metacele, that is absent in fish. In elephant fish, Mormirus, the cerebellum is very well developed and is probably associated with the reception impulses from the electric organ of fish. Prominent irregular projections called the auricular lobes or restiform lobes present in certain fishes, such as Koalas acanthias, and continuous with the medulla oblongata are actually parts of the cerebellum, and these are the center of equilibrium. The body of the toad and frog is broad and less weight, already in stable equilibrium, and for these regions our cerebellum is very poorly developed. The greatest advance in the cerebellum of the higher forms is the appearance of a pair of floccular lobes near the ventral sides. They correspond to the auricular lobes of fishes and first appear in the crocodilians. In birds and mammals, the cerebellum consists of a prominent middle portion, the vermis, with floccular lobes on either sides. The cerebral cortex of the cerebellum that covers the white matter and that show a complexity branched arrangement that is known as arbophyty. The cerebral cortex consists of the three regions: the deep granular layer, that is a granular layer you see here, that contains several types of cells. It's a middle layer, that's middle layer is a Purkinje layer, and the superficial layer, that superficial layer is a molecular layer that consists of fibers and synapses. In mammals, the vermis is divided into anterior, middle, and posterior lobes. The bilateral extension of middle lobe are cerebellar hemisphere. The surface of the cerebellum is thrown into numerous folds of gyri, and that are separated by the deep grooves that is known as sulci. The ventral side of the metencephalon is marked by a conspicuous pons that is composed of prominent mass of transverse nerve fibers. The last one, myelencephalon. The myelencephalon, that is the last part of the brain. The medulla oblongata is situated beneath and behind the cerebellum and continuous to the spinal cord. The lateral and ventral walls of the medulla thicken, but the dorsal wall retains its epithelial character. The pyometer, a vascular membrane surrounding the brain, fuses with the brain lobe. Vascular folds of tela choroidea extends into the fourth ventricle to form the posterior choroid plexus. The thickened ventral and lateral walls of the medulla contains large white fibered tracts as well as several columns of gray matter. In higher vertebrates, the integrity of the columns of gray matter in the brain is lost and a separation into various nuclei has occurred. The nerve 6 to 12 are associated with the medulla oblongata. It contains important nerve center which control regulation of heartbeat, respiration, and metabolism. In fish, medulla oblongata is broad. That is the medulla oblongata. In front and has one medial facial lobe. If you see that of the facial lobe, and you see here that of the vagal. The vagal lobes are just at the center of the medulla oblongata and are better developed in some species. That's depending upon taste for feeding. Facial lobes are prominent in species that search their food, exploring the surrounding with the help of barbels, such as celluloid group of fishes, Mr. Vitatus, Sylviid fishes, Eutrophis bacha, etc. The dorsal anterior portion of the medulla oblongata, that is referred as acoustico-lateralis area. And this area contains nuclei associated with nerves from the lateral line system and the inner ear. In terrestrial vertebrates, it is associated with the equilibration and auditory function of the ear. These are the reference books: Elements of Chordate Anatomy, Weickert; Comparative Anatomy of the Vertebrates, Kent and Miller; Integrated Principles of Zoology, Hickman et al.; Vertebrates, Comparative Anatomy, Functional Evolution, Cardon; and the last, the vertebrate body, Romer. And the figures that are mentioned in my presentation, some of the figures are also captured from the Google image. Thank you, everyone.